Okay, so let us get it going. There is some echo somewhere. Any? Maybe you have to mute. Mute. Speaker. Hello. Thank you. So, good evening and welcome to today's ASET colloquium, which is titled as Understanding Behavior and Disorders Using Brain Imaging Data. Obviously, very interesting and important topic of today's research, spreading both into life sciences and also using machine learning and other techniques. We have Dr. Kaustubh Patil. He is very kind to give me the English version of his institute, so Research Center Juelich. He is going to speak today on this very important topic. Before I go and formally introduce today's speaker, I would like to thank Mandar. Mandar is here, who was instrumental in bringing this speaker here. I will say a few words about today's speaker. So Kaustubh actually received his bachelor's degree in electronics engineering from Shivaji University in 2003, and then went on to pursue his master's in artificial intelligence and intelligent systems from University of Porto, Portugal. And then he, of course, which he completed in 2007 and did in 2013, he received his PhD degree from Max Planck Institute of Computer Science. After completing his PhD, Kaustubh worked as a postdoctoral fellow at both University College London and MIT between 2013 and 16. And then he joined where he is right now working, Research Center at Juelich in 2017. He now leads there an applied machine learning group. His group actually mainly focuses on designing and evaluating machine learning algorithms and pipelines to better understand biological systems, and in particular to those related to mental health and psychiatric disorders, as well as basic understanding of brain structure, function and development using magnetic resonance imaging data. So that's what I said. The topic straddles to many fields, both applied as well as basic. And I think we can really look forward to very interesting lecture by Kaustubh. So with these words, I would like to call Kaustubh here for his colloquium. Thank you. Yeah.
Can you hear me okay? Thank you. So this is the first talk, external talk I'm giving after Corona times. So I hope it goes okay. See, I need this. Thank you very much for kind introduction and also for inviting me here. I was here a few years ago visiting Mandar, but this was already like about 10 years ago. And I see that a lot of things have changed. The campus looks quite different, I think, and better even. And I will put this here. I was told not to move it too much. Okay, seems to work. Thank you. So just a simple introduction slide, right? So why do we want to study the human brain? Right? It's one of the most complex organs in the human body. As we all know, it has billions and billions of neurons. Some estimates say somewhere close to 100 billion neurons. And there are trillions of connections. So it's extremely complex organ. And it gives rise to most of the behavior, at least that's the presumption. So that's the dogma in the neuroscience, right? So everything, these neurons, the connections, how the neurons talk to each other, it gives rise to all the behavior we do. Every decision we make, every action we do is somehow controlled by the brain and the nervous system that comes with it, right? So that's why it's one of the interesting things to study, but it's extremely complex. So how can we go about this, right? So of course you can go at the very molecular level and study the neurons and the gates, that how kind of neurotransmitters kind of go back and forth, or one can also somehow take like a macro level. The good thing about this macro level is we can study this in vivo using MRI. What does this mean? It means it's non-invasive. So we can take patients, we can put them into MRI, we can see what's happening in the brain. And that can actually help patients in vivo. So that's one of the main reasons why MRI has become so popular over the recent years. What we do in my lab and in the wider institute in the research center Juelich and the connected sister institute at the University Hospital in Düsseldorf is study the behavior and also how the behavior abnormalities, for example, in psychiatric disorders, and in some cases, how neurodegeneration happens using MRI data. And we specifically combine this MRI with machine learning. And I will try to talk how kind of we do this. I'm sorry that I'm going to glance over many, many details. I will try to give you kind of a broad perspective, but if you want to know any details, I will be more than happy to discuss them. Okay. So a very quick slide about what type of data we get from MRI, right? So I don't need to tell you about physics. I know very little, very basic physics of MR, but very simply spoken, what we do is we take a 3D pictures of the brain, right? And it's basically converted into what we call as voxels, is a 3D pixel, right? So it's called a voxel, and we have hundreds and thousands of voxels depending on the resolution. And what's interesting is we can get something called as the structural image. So this is in MR language, it would be called T1-weighted image. That tells you about different tissue types that exist. So there is gray matter, there is white matter, and there is CSF. These are kind of three basic fundamental tissue types. So that's why we can study them using structural MRI, but there is also something called as the functional MRI images. So there is no time component in the structural, right? So it just tells you the composition or the structural organization of the brain at that moment. That's all. But in the functional MRI, what we do, you take multiple images in sequence. So there is a time component to it. And then with that information, you can start to study how the different regions talk to each other. And I will try to give you examples how they can be used in the both cases. So we will first look at the structural MRI, and then we will move on to the functional MRI. Okay. They have slightly different uses. So this is not a very strict differentiation, but just to kind of orient ourselves a little bit. So usually structural MRI is useful to study aging or neurodegeneration because it's somehow structural changes that happen in the brain, right? And functional MRI many times is somehow considered as the connectome of the brain, right? So how different regions talk to each other or how they're connected, functionally connected to each other. There does not need to be a connecting wire, kind of axons going to neurons, but they still can kind of behave in synchrony to each other. And that many times is useful in looking at the behavior, because there is some time component to it. So the brain kind of dynamically needs to create behavior, and that's why you need to study the time dimension using the functional MRI, right? And this is, again, many psychiatric disorders, for example, schizophrenia. It's called as the disorder of connectivity, right? And that's, there are several reasons because, again, many behaviors change in these disorders. And I will also try to give you one example of that. Okay, let's look at the structural images first to get you a little bit excited, right? So just as a guess, one structural scan, a standard resolution scan, takes somewhere about 5 minutes. Imagine we put you in the scanner, take one scan in 5 minutes. Can it tell how old you are, right? So that's the question. Just try to guess. Maybe you think yes, maybe you think no. Maybe you already guessed the answer if I ask a question like this. But let's say we have a big database of many people from young to mid-age to old, a big database like this. We can train a machine learning model. So we know the MRI images of those people, and we know how old they are. So we can train something called as a supervised machine learning model. And once we have that, then indeed we can ask the question for a new individual for whom we do not know how old that individual is, how old this person, simply using their brain image. I will explain why it makes sense, or because we know the age of almost any person, or we can just ask them, right? So why does that make sense? We will come to that. But indeed, it's possible. So what you see in that scatter plot on the x-axis is the true age or chronological age. On the y-axis is the predicted age using the model, right? So you can see this kind of line. It works quite well, right? It's not exactly 100% accurate, but okay, it works well. And the difference, usually the difference between the predicted age and the actual age is called something as the delta. And the brain age delta is useful for multiple reasons, right? So what you see there, let me try this. Now, if it works, okay, here. What you see, this 1 point here, right? This brain is the predicted age is much higher, let's say somewhere close to 45 or so than the actual age. which is somewhere close to 20. So
From the predicted data from the error, how does the scatter of the training data?
of the training data.
Get the error?
So you mean yes. So you mean the training points, how they would look like? They, yes. So I'm not sure if I completely get your question. So if I would use like the training data to plot this, because this is on the test data set, right? If you could create exactly the same plot on the training data, it would be much accurate, much more accurate. Yeah, it will be much closer to the line. The spread would be lower, right? On the training data set. Okay. Any other questions? Yeah. Yes, exactly. That's correct. So it's a regression model because it's a continuous outcome, right? Okay. So what's the use of this model is now this one particular individual. Let's see if I can find the pointer back somehow. No. Maybe if I do it here. Okay, there we go. But this is not working now. It works, but it moves very fast. Okay, I will go back here. So here, this data point there, right? So the predicted age is 45, or somewhere close there, and the actual age is 20. So what the machine learning model is telling us, this brain looks much older than what it should look like, right? And this is useful, again, because it can be indicative of abnormal aging, because the brain is aging much faster than the time, or at least the age of the person, what we should expect from this. So that's the signal. It's called the delta. And in many cases, brain changes happen many years before the actual external symptoms come about. For example, if you think about Alzheimer's, right, or some kind of dementia. So we notice it when, let's say, there is some kind of memory loss or so on. But many years before, 4 to 5 years before that, actually the brain has already changed. So the external symptoms come much before, much after the brain changes have actually happened. So some model like this can be used as some kind of early warning system. If it says something like, okay, this person's brain already looks older than it should, it's kind of a red flag, right? So we can say, okay, hey, clinician, pay attention to this. There is a question?
And do they eventually get some
Yes.
How do you look at something like this and differentiate between when the model is giving you some real value versus when the model is just making?
Very nice question. Very good question. Thanks for that. So I will just repeat the question in my own terms. So hopefully it's easier for me to answer the question, right? So the question is basically, what's the difference between the model always has some error? There is always a modeling error. So how we differentiate this modeling error from the actual error that we want to capture, the biological difference versus the modeling difference, right? So that's the question. It's a very nice question. And of course, every error is a mixture of these two components. And I will show you some examples where this difference actually shows up in disorders. It has some kind of signal that we can capture. So of course, the modeling error is never going to go away. Maybe in the future it does, but currently it's very difficult, right? Sure. Yes.
But there are a couple of mics planted.
Hello. How many biological samples were used for the creation of this data set? And what is the ethnicity used? And what is the scalability of this to different races and different genders and stuff like that?
Yes. So thanks a lot. Again, very nice question. So I like that a lot. So for this particular plot, I don't remember exactly what was used, but the paper that I will show you afterwards, for the training, we used somewhere close to 3,000 individuals for training the models, but now we have more models, new models with about 10,000 individuals or so. The next part of your question, they are mostly, if not all of them, are Caucasian, because the data was collected either in Europe or in the US. So the most of them are Caucasian populations or cohorts that we have. Unfortunately, that's the currently dominant data that's available. And I will show you as currently as a minor point, but I will come to that afterwards again. And in this case, we actually check that there is a reasonable balance between males and females in this data set to kind of have some good representation of both. So we also try to build models for them separately, but at the end, we decided to go for a single model because they did not show much differences. But we have other projects where we actually try to separate male and females, right? Because it's also interesting to know where if there are differences, and if yes, where they're coming from. Yeah, we have found both structural and functional differences, but unfortunately, I don't have the slides on them. I did not include those for those.
Yeah, so maybe some pointed questions here and there, but let the speaker move, and we have time at the end for questions. But don't get distracted, it's not talking questions. Yeah.
So, and one nice thing about a model like this, it's called a normative model. And what that means is, in this case, it's kind of disorder agnostic, because we do not need data from a particular disorder, which many times is very, very difficult to get. Right? Just as an example, so many patients, especially in advanced state, they move quite a bit. So it's very difficult to get kind of good quality MR images from them. So in this kind of models, we don't need data from patients because it's a normative model and we can use it in disease agnostic way. And that's kind of one of the appealing points. Again, how exactly and how well it works, does it make to clinic and so on? These are different questions, but at least we are trying to make one step in that direction, right? Okay. Again, I'm going to skip that just by saying this is what I mentioned. There is kind of 3 tissues that we can extract from MR. There are possibly more that can be done, but this is kind of the dominant way of extracting. And we are focusing on the gray matter here. It's difficult to use for me. So we are going to focus on the gray matter. And the reason being that's where most of the neurons are. And again, the neurodegeneration often happens in those parts. And that kind of gray matter is also where deep brain stimulation kind of usually happens. So that's partly the reason why we focus on the gray matter. Of course, other tissue types are very important. There are many studies that show that, again, aging signal is there in other tissue types, but in our work, we mostly focus on the gray matter volume. Right. This is a pipeline. Again, we don't need to go through all the details of what exactly happens, but given an MR image, we need to do quite some processing until we can start using the output of that image, because the raw image by itself is not very useful. So we kind of go through a pre-processing pipeline. What I show here is something called as the CAT toolbox pipeline. So it's a very nice out-of-the-box pipeline. You can just throw one MR image at that, and it will do the tissue segmentation, normalization, all the steps, and it will say, okay, here is the gray matter volume for each voxel in the brain, right? So it gives you some kind of voxel-wise output, but there is a question, right? So what do we do with this voxel-wise output? There are plenty of options. Again, for any machine learning modeling, we need to make many, many choices, right? So in this such a high-dimensional data, for example, if you have a high-resolution image, you can end up getting hundreds of thousands of voxels and only a few thousands of subjects. So that is going to be bad for the machine learning algorithm because it's going to overfit, right? So and that's kind of a problem. So we need to extract some features that we can feed into the machine learning algorithm. So these are the choices. Some of the choices that are listed there. You can go, for example, there are kind of predefined parcellation schemes of the brain. So you can divide the brain into some kind of regions and take the average within each region. Or you can resample the voxel. You can make them bigger, kind of some kind of linear or nonlinear transformation. So these are the choices one can make: apply smoothing, no smoothing, and so on. Plus, there are many different machine learning algorithms one can choose from. So all this space is very complicated to kind of decipher. So we need to make choices how to do this now, right? So actually get a model that's good. Good meaning highly accurate. It's not only accurate, but there are many other things we need to and want to consider, right? So to have a good model that is reliable. So we don't only want a model that works in some specific scenario, but we want a model that generalizes. Let's say you have multiple clinics, and there is some data that the model has never seen from a particular clinic. Now, if you know the MRI scanners, they have very specific idiosyncratic noise introduced into them. So there is some kind of systematic noise. If the model has never seen that, it might fail. So we want to make sure that these models kind of work within site data, so where the models are trained, but also on the data that the model has never seen from a new site. So it should also work cross-site, for example. But also we want it to be reliable. What does that mean? If you scan the same person twice within some reasonable amount of time, we don't expect the brain structure to change much. So the predicted age should be very similar in that case. That means the model is reliable, some kind of test retest reliability. And the third case, the third point here, we hope that the model is also longitudinally consistent. And what I mean by that, if you scan the same person twice, but few years apart, right? We expect the model will predict some kind of proportionally higher age. If the aging is in some kind of normal, is following some normal trajectory, right? Then we expect that. Does that really happen with all these models? So these are the questions that we asked in the specific paper that I cited there. It just came out a couple of months ago. How did we do this? So we evaluated 128 workflows, which is combinations of different feature spaces that I mentioned. So some kind of parcel-wise features or some kind of resample voxel-wise features, and 8 different machine learning algorithms that have been used for similar type of modeling before, and also some that have not been used. So we put all of this together and we followed some kind of a funnel pipeline. So we sequentially reduce the number of models we test to making for the models to kind of pass some kind of stringent criteria. And hopefully we arrive at a single workflow, which is very good in all these cases. So what happens, right? So these are the results of what we found. So again, this kind of jargon there, right? So S4R4, it just means we smooth with 4mm kernel and we resample to 4mm voxel. So this is some kind of pre-processing step that can happen, and we still have the voxel-wise output. So that turned out to be the best kind of feature representation in our evaluation. So what you see on the right-hand side, this kind of density plot, it's not exactly density plot, but it's a ranking of the models. It's 128 on the top left, and then the 32 models on the top right. And what we always saw is the Gaussian process regression and also relevance vector regression. So these kind of 2 models ended up being very good in all these different scenarios. But there is, as always, it's a research question, not everything works that well, right? So we managed to get quite nice kind of within cross dataset generalizability. The reliability was also quite nice, which I don't show here for brevity, but the longitudinal consistency was a problem. So what you see in these 2 scatter plots is 2 data sets where we have data from about 3 to 4 years apart from the same individual. So it's a longitudinal data. And in the first data set, we see a positive correlation. If the true age increases, the predicted age also increase in some kind of a linear way. But in the second data set, it did not. So the longitudinal consistency still remains a problem. But the model, is it still useful? And that was the previous question. What exactly is this delta, the difference between the true and the predicted age is telling us? Is there some biological signal behind this, right? And this is a lot of plots. So I did not realize that before, but I will try to explain very quickly. So the most important plot is the middle left. Let's say if we focus on that plot there, if I can find the pointer like this part here. So you will see 4 box plots. The first one is the healthy controls. Then the yellow one is the early MCI. So that's the mild cognitive impairment. So this is considered some kind of a pre-stage to possibly moving to Alzheimer's, right? Then you have late MCI, and then you have Alzheimer's. What you should see is it's linearly increasing trend. And this is the delta. So this is the difference between the true and the predicted age, but using the same model that I showed you. And this data set was never seen by the model itself. So this is a completely out of sample prediction that we have. And on the right, middle right, it's a time point 2. It's the same subjects were scanned again. So we kind of see the longitudinal consistency, and even the delta goes up a little bit, and it should, especially in the Alzheimer's, where the neurodegeneration is happening relatively quickly. What you see in the bottom, the scatter plots, is the 3 kind of clinically used scales in diagnosing Alzheimer's. And we see that the delta also correlates relatively okay. It's not, of course, not a super high correlation values, but it still correlates to somewhat degree to this clinical scores. So we think that, of course, it's not perfect, but there is some potential of these type of models. I hope that makes sense. I'm going to move on to the final slide of this part of the brain age prediction part. And of course, the applications are Alzheimer's, but the problem again in machine learning is how do we exactly do a model selection? And what does that mean? Of course, we tried 128 and we put accuracy as our goal. We want the model to be as accurate as possible in these all different scenarios, right? but that might not be the right way to do model selection for this particular task. It might, some lower accuracy model might be better because it captures the biological signal. So that could be the case. So we are currently working on figuring out if that is the case or not, and if yes, how do we find it out? Of course, other disorders, one interesting project is we are applying these models to astronaut data. And many people or clinicians are interested how the brain changes because the gravitational pull changes, right? When people go into outer space, when they come back, and there are some physiological changes that happen for sure. But does the brain change? Does the brain aging change? So that's an interesting question, and we are looking at with some collaborators on astronaut and cosmonauts data as well. We also have some methodological projects going on. How can we improve the model accuracy and interpretation as well? So these are kind of, let's say, more going into the direction of methods development in the core machine learning. So we are experimenting with some stacking type of models, but we are also moving towards more kind of deep networks with specific loss functions to make them rank consistent and so on. So this is just kind of, this is what's coming, let's say, in a year or two. So that's where we want to go. So I hope at least some of this makes sense to you. And I'm not sure how much time I have. Okay, good. So, okay, thank you. So any questions about the brain aging part? Maybe it's a good time before I move on to the next section. Okay, I don't see any, so okay. So I will move on. So yeah, sure. That, yeah, so let's say an easy thing. Again, it's a very interesting question, but difficult to answer because you're asking kind of similar question: what is the model error and what is the biological signal? The balance between the two, right? So if we look at the modeling error, so what we see is somewhere close to 4 and 5 years. So if the deviation is within that, it's going to be very difficult to say. But anything beyond that, I would say it's going more towards the biological signal. Another complicating point is the pre-processing, because sometimes it does not work as well on some images because of some artifacts or some kind of acquisition problems. And then, of course, you get kind of very weird outliers. So in this case, we just look at them manually. There is at least no very kind of well-established automated ways for doing that. I hope it answers your question. Yes. Okay. So yeah, sure.
The outliers that you see, if I'm assuming you are participating with clinical data, and so like to the outliers that you are seeing with your have people seen actual behavioral changes or actual clinical changes, and how well does that correlate to change in the predicted age?
Yes, so what I showed you in the previous slide, let's see if I can go back. Yes, so these are the behavioral changes in the subjects. So these are behavioral scales. It's very small to see, unfortunately. But for example, the first one is the mini mental state examination. So this is very commonly conducted test in this domain, and we see a relatively nice correlation. So the R is negative 0.45. Which in a clinical domain is quite nice, actually. So we were very happy to see this level of R value. So there is some signal in there in terms of behavior as well, because this correlation knows nothing about itself about the aging process itself, right? So all it knows is the delta, the difference, and the behavior itself. In some other cases, it can be a little bit complicated. I do not have slides ready, but we have some interesting results on a sample of schizophrenics. And in that case, we see much higher correlations with behavior. So it again depends on probably many things, right? Like how well the data is matched to the training sample, the composition of the distributions of the train and test data, and so on. But in general, yes, we see some decent correlations between behavior and the delta, the brainish delta. So this is an ongoing work. It's a disclaimer, right? So we know some things, but we don't know many things about this project. But I also wanted to kind of give you some idea about new projects and not only something that we have already resolved, right? So it's a little bit exciting to talk about those things. So in this case, there are, again, many different ways one can look at the MR image. So one is kind of this intensity values, then you can segment, you can find the tissue types, and then do kind of this voxel-wise intensities and so on. But in this project, what we are looking at is the shape of the different brain regions, right? So if it's kind of a signal that has been not well investigated yet. And our hope is the shape itself can tell us many things that other measurements cannot tell us. And in this project, we are using data from UK Biobank, which is about 20,000 individuals. So it's a very, very big data set. So we have selected a subset of about 40,000 individuals, which we consider with some criteria as healthy sample. And the goal is applying something called as the shape DNA, which was proposed in 2015 by one of our collaborators, that applies some kind of Laplacian transformations to the shape. It first builds a mesh using the triangulation mesh on the locations of different voxels of that shape. And then you apply a Laplacian transform and just calculate some eigenvalues, let's say using SVD. And that describes the shape reasonably well. And that was shown in 2015. And we are using that to actually see if there is a genetic signal behind this, because if it's inheritable, we think that it's more likely that we will find also some application to different disorders. So this is the first step we are doing. Because it's a UK Biobank data, we not only have MR images, but we have lots of SNP data as well. So we can actually run GWAS studies on this and try to find out if there is some kind of biological signal. So I will show you some preliminary results. As I said, these are kind of relatively new. So we know certain things, but not all of them. So this is just an example of how the brainstem looks like for 3 different individuals from this UK Biobank data. So just looking at it, we can see like, okay, there are certain differences, not only the size of it, but there are also differences in the shape of it. So this is the first results of the GWAS. And if you're used to looking at this kind of what are called as the Manhattan plots, you should see that there is some signal, which is quite strong. So the higher the points go, the stronger the signal is. So it's kind of negative of the p-value, kind of log the p-values. That's what we are looking at. And why this is interesting is because then we can go back and try to find out these locations, the SNP locations, the RS locations, where are they located in the genome, and try to find out which genes are related to them, and then further go on and try to find out if these genes together that are significantly showing up in this kind of GWAS analysis, if they're part of some biological processes. Okay? So this is relatively standard bioinformatics pipeline, but the input is something that no one has looked at so far. So this kind of shape of the brain regions. And what we see in this case is, of course, we will see some kind of physiology-related genes, which is always the case because the genes kind of contribute to many, many different biological processes simultaneously. But interesting is we also find something that's related to neurological processes. For example, the reaction time and neuroticism and Alzheimer's. This is something we think is very exciting. Right? So if the shape turns out to be a good marker that can be extracted from MR image, and it also shows some genetic component, then we think it has the potential of kind of moving it towards a real biomarker that can be used into clinics. So that's again the goal. So we are making some steps in that direction, and these are some preliminary results which I hope kind of make sense that we can pursue this direction a little bit. Just to give you a summary, so I understand that it's not very easy to read, but I just wanted to kind of advertise or show off a little bit. So this is structural MRI plus machine learning is kind of one of the core points of what we do in my lab and also in the wider institute. And these are all the papers that came out relatively recently, either by the first author being the member of my group or some collaborators that are kind of using the similar framework that we are using. And there is, as you can see, there is some kind of interest in sleep research. There is stroke data in there and so on and so on. So there is many different applications that we can find once we establish a framework and pipeline like this. Okay. So I will skip the summary a little bit, and we can move on to the functional MRI, which is also very exciting because it looks at a very different type of information that, again, we are extracting from MRI, but now we have a time dimension in. So instead of taking a single image, we take many, many images. The bad kind of thing is the resolution, the temporal resolution is higher. Of course, it's more than one, but the spatial resolution goes down because we are taking many images. So they're relatively noisy in the spatial dimension, but we're interested more into how the different brain regions communicate with each other. And what I mean by that, again, I will skip this. I'm sorry, I think I forgot the actual order of the slides. So I will go front and then I will come back. So what's happening there is a time dimension. So again, we take many, many images and then we apply a parcellation scheme to it. What does that mean is we take a certain voxels, which are somehow close proximity and have been predefined as a brain parcel. And we just average all the time series. And that gives us kind of a region-wise time series. And we extract that kind of time series from all the brain parcels. Again, let's say parcellation scheme has 200 parcels, right? So then we have 200 time series, and then we calculate all pairwise correlations. Again, the standard being the Pearson's correlation. Yes? It, again, depends on the scanner, but the standard is somewhere close to, let's say, 2 seconds. And there are some data sets that are, they're rare, but kind of becoming more and more common with some kind of better sequences that are somewhere close to 0.7 seconds, so even less than a second. So again, the temporal resolution is not as good as MEG, let's say, or EEG, but the spatial, there is a reasonable balance between the spatial and temporal resolution, right? Yeah. And once we calculate the connectome, which is the Pearson correlation, then we can use that information, trying to see how communication between the different brain regions, is it affecting behavior, is it affecting disorders, and so on. So this is in a step towards creating a feature space that we can use for machine learning analysis afterwards. Okay. So with that, I will come back a little bit to explain what we are doing. Yeah. So there are traditionally two different types of fMRI. So one is called as the task-based fMRI, and the other is called as the resting state fMRI. So in the task, there is some kind of explicit task. So for a standard one being finger tapping task. So there is a screen in the scanner, and some instruction comes like tap. And then all the participant needs to do is to move their finger. So they're told exactly which finger to move, for example, the right hand index finger. So this is kind of a finger tapping task, right? Or there are kind of, can be more complicated behavioral or cognitive tasks. For example, you need to remember some sequence of letters and need to recall them and so on. So that is called a task-based fMRI. The nice thing is, because you have a very specific hypothesis about if you do this type of task, you should see some activity in the brain in certain regions. So that's how these tasks are designed usually. But there is also something called the resting state MRI, and in which there is no explicit task. All the instructions the participant has, please lie still and try not to fall asleep. That's it. And that already gives you quite nice signal how different parts of the brain regions kind of in a way communicate with each other. And the reason I say communicate and not connected, because there is usually no kind of a direct connection, at least if we see some kind of diffusion imaging, but they kind of go together with each other in a synchrony, right? So there is some kind of communication happening. That's the hypothesis, right? The good thing about resting state is you do not need any instructions, so it can be used in participants that can have difficulty focusing on a task. So again, in clinical populations, this can be very useful. So this is one of the classical output of a resting state brain scan, and these are canonically defined resting state networks. So what you see here, these different colors and different kind of jargon, probably for you, these are different networks that have been defined on thousands and thousands of individuals that these brain regions kind of talk to each other. The similar color means they kind of go up and down. It's a correlation at the end, right? And for example, the DMN, which is called the default mode network, shows up everywhere in the resting state analysis. And that has created many, many hypotheses of how disturbances to this specific network can lead to different disorders. Just to give you an example. And this is called as the intrinsic connectivity. And as I said, it can be very useful in certain cases where the task engagement is not possible or is difficult. So I will skip this. So once we have that, like a big connectome, right? So let's say even with simple 200 nodes, I'll try to get the cursor there, we will get somewhere close to 20K connections. So this is a huge feature space. And many times machine learning algorithms are not happy if you do that. So what we came up with is some kind of biologically meaningful priors. So can we put priors on this connectome that we have learned from the literature to kind of reduce this dimensionality? And what we do in this case is select a subset, but how do we do this is something called as the meta-analytical priors. So let's say people have kind of studied before a particular task engagement, let's say some kind of n-back task where the working memory is involved. And we know in certain disorders, this working memory might not be functioning as well. In that case, we can use this working memory network as a prior to not to use the whole connectome, but just a subset of it. Right? And once we have this kind of, so we have many, many different meta-analytic networks that people have studied over the decades. So it's basically reusing the knowledge that others have created at the end. And this is the outcome of one of the PhD students' work. So what you see on the left-hand side is, again, 16 networks. These are 16 meta-analytic networks somehow shown in how the different nodes are and the connectivity between the nodes. And using this connectivity, we try to predict a scale or kind of factorized scale of schizophrenia patients. So this is called the PANSS scale, which is very standard kind of used in the literature and also in clinical cases. But what we found in the paper that I said 2020, so these are two different papers actually, 2020 and 21. So in 2020, what we did, we factorized this PANSS. These are 30 items in that scale. So we factorized this using something called as the non-negative matrix factorization. If you have heard this term, so it's some kind of factorization, which is kind of like, let's say, PCA, but the output is always non-negative. So it's kind of has more interpretability, and it also tries to separate the different factors cleanly from each other. And this is what we found. So instead of predicting the PANSS outcome, we try to predict our own factorization. And we saw actually our own factorization can be predicted better than the PANSS scales. So we applied the 16 networks to predict these 4 outcomes that we had from our factorization. And then, of course, we applied Bonferroni correction to make sure that we are not kind of over-interpreting our results. And what we see as an outcome of this is, again, a little bit full slide. But we saw that the ESAD network, so as it's written there, is socio-affective default mode network, it predicts the cognitive symptoms. So one of the scores can be predicted relatively well with this. Again, what I mean by relatively well is correlations of somewhere close to 0.2, 0.3, right? But this is, again, in the clinical domains, the biological systems are really, really messy usually. And especially in clinics, they get even messier in disorders. So when we get correlations like that, we are quite happy. Then we actually replicated our results in 2 cohorts, and we saw, again, the same results show up. So we are happy that there is some kind of reliability to the results, right? And then to make the next step, what does it exactly mean, right? So why are we seeing this outcome? Then we went onto some kind of PET imaging. So this is a neurotransmitter and receptor data, that what you see on the right-hand side, top right. And we extracted the node weights. So how important each node is in predicting the outcome. And we correlated the spatial value of this weight to the spatial value of different neurotransmitters. The hope is, if we see a good correlation, then we can actually go one step beyond and say it's not just the connectivity, but it's somehow driven by the neurotransmitter receptor system in the background. And nicely, what we saw, FDOPA and serotonin receptors actually quite nicely correlated, and also significantly after correction with the values that we had. So that's the outcome of, again, a PhD student's work. Actually, I showed that before already. And that's something that we are actually pushing more and more in this direction. So actually to kind of bridge these different levels of interpretation, because the brain system, of course, we think of neurons and so on, but neurotransmitters and receptors play a very, very important role. The problematic thing is getting the data. So usually, because it's a radioactive material that you need to inject, it's not easy to collect that data, and even in many cases not even ethical to collect that data. So most of that data is normative. So it's not possible to get a very big databases like that. So we kind of average across like dozens of subjects to get some kind of normative maps from this. And a final project that I will show about this connectivity between different brain regions is whether we can find signals for behavior, right? And this is something very interesting. So there is no kind of clear disorder angle in this, but just understanding how does brain or the connectivity between different brain regions give rise to behavior. And this is a very important question, but very difficult to answer because it's an extremely complex system. We don't even know which features are meaningful there. So based on the 2020 paper, we had the hypothesis that there might be some time points in this time series. Let's say we have a time series with a few hundred time points, and we usually use all of them to calculate the connectome, right? But from this 2020 paper, there is some kind of hypothesis that says that this might not be the case. There are a subset of time points which might give you at least similar or even better information about behavior. So that's what we wanted to test. So we followed their pipeline. Again, I'm skipping quite many details exactly how it is done, but what we get at the end is a ranking of the time points. And on one end, you have the high amplitude co-fluctuation, so where many brain regions are somehow going up and down together. And the other end, you have low amplitude co-fluctuation, so where they are kind of more with asynchrony with each other, right? So you get a ranking like that. And then what we can do, we can sequentially go from the one end to the other end, or the vice versa, right? So this kind of two directions, and we calculate the connectome with this subset of time points. And using that, again, we use the machine learning kind of standard pipeline to predict behavior. And I will show you what we get. So again, if we focus on the left plot, we try to predict crystallized cognition. And crystallized cognition actually is relatively easier to predict because this is something that gets encoded in the brain. Many studies have shown that. And what we see if we start from, so the red one is the high amplitude co-fluctuation. So we are going from the high amplitude to low amplitude, and the blue one is the other way around. So we take a few time points and then we concatenate them. And then we calculate the connectome using those as features. We try to predict cognition, right? And as we increase the number of time points, and the reason why the threshold to 50, because up beyond 50, again, we kind of crossing the directions, right? So that's why we stop at 50. And we see we can predict with correlation somewhere close to 0.45, which is not too bad in a data like this. But as we move towards right, you see and the dashed line is the full connectome using all the time points. So as we sequentially add the time points, we reach the full connectome. And as we move towards the right spots, we see the prediction itself goes down. And this is quite a common case. So of course, not all behaviors can be predicted using the same data. Of course, I wish and it would have been very easy if they would, right? If we could predict all the behaviors using the same data, it would be a reasonably solved problem, but unfortunately, that's not the case. It's also good because then I still have a job, right? As we move, and that's kind of, we are wondering again and again, why that's the case? Because on one case, we can predict crystallized cognition quite nicely, but the fluid cognition, to the right-hand side, we cannot predict, at least almost nothing, right? And these are two components of intelligence that are very frequently studied in the psychology literature. Of course, they have different meanings, but we would have hoped, or many people hope, that at least we can predict both of them reasonably okay with similar type of features, but unfortunately we cannot. And interesting point is the original hypothesis that maybe there are some subset of time points give better information did not hold. So we could not confirm that hypothesis with this study. And this paper is currently under revision, so hopefully it comes out soon. Again, a little bit of advertisement. Similar to the structural MRI framework, the functional MRI framework is also quite interesting and can be used in many, many different domains and many different directions. So what I showed you is just a subset of what our group and institute is doing. And these are, again, some examples of the things that have come recently or will be coming shortly. And one interesting thing is the top right, where people are watching movies, right? So they're not just laying still in this case, or they're not doing explicit tasks, but they're shown a movie. And the hypothesis there is it has a better task engagement and also engages many different subsystems of the brain, because different types of movies hopefully engage, let's say, emotional or fear networks and so on and so on. So the hope is like we can kind of quantify the connective or the communication between the different brain regions better using data like this. So that's also something. The data is being collected in our group or our sister group at Juelich Research Center, and we are lucky enough to have access to it, so we can try things like this with that data set. And finally, I would move on to the confound leakage. So this is kind of, I'm moving a little bit more towards machine learning because I thought that would be of quite interest. So I'm not going into too deep into machine learning, but I will try to focus on one specific type of problem or pitfall that we recently found. We spent quite some time understanding it, but now I think we have some decent understanding of why, what's the problem and why it is happening. So I will try to convince you, and let's see if you think it's the same or not. Okay. But I can make a pause for the questions on the previous part, if you like. Yes. 28. Okay.
Some of us don't understand what the last two plots mean.
Uh-huh.
What is cognitive flexibility and fluid cognition?
You mean these two?
Yeah.
I
We
see.
just
Okay,
understood
good. So the 4 plots are different cognitive measurements. So we have a data set where X number of individuals, in this case, somewhere close to 800, I think, they were scanned, and they were also given some standardized questionnaires outside the scanner, right? And these questionnaires give you a score. So for example, what's the crystallized cognition score? What's the fluid cognition score? What's the cognitive flexibility score? So these are a kind of standardized scores in the psychology literature. So what we are trying to do here is we take the functional connectome and try to predict those scores one by one. So in the first one, we predicted or tried to predict the crystallized cognition. In the second one, vocabulary comprehension, then the cognitive flexibility and fluid cognition. The reason why we arranged it in this way, because the accuracy goes down. Right? So that's the order that we decided.
So I just wanted to understand the layman meaning of the terms cognitive flexibility and fluid cognition.
Okay.
The first two I can guess sort of.
Okay. So the fluid cognition is something that kind of, let's say, more dynamic thinking. So it's not something that you learn through the education system. So it's a little bit more abstract. So that's the fluid cognition. So let's say kind of mental rotation tasks and so on. And cognitive flexibility is how quickly you can change between different tasks, so different types of tasks, because different kind of psychological systems need to be engaged. So if you switch between them, there is some kind of tax involved in switching. And the cognitive flexibility tries to measure that a little bit, like how kind of nicely you can switch between the tasks. So I hope that, again, this is not like a very detailed explanation. I'm not a psychologist, right? So I know what I know from the colleagues and when I read the papers. But if you want details, we can, of course, try to get into it.
Thanks. Thanks.
Yeah. Okay. This one or before?
No, previous one. There was a
This one.
Yeah.
Okay.
Network node importance correlate. How did you get that basically?
I see. Okay. So in this case, for example, we have the connections that we know end up being predictive, right? Because in this case, we used, if I remember correctly, we used the relevance vector regression, which has with a linear kernel. So it has a decent interpretability. So it tells us which edges have a non-zero weight on them. Okay. So we extract those edges, and for each node, we just simply add up the corresponding weights for each node of all the edges connecting it. So it's some kind of a degree centrality.
Can they be reliably traced back to the features that you see on the? So by receptor densities, you mean just the nodes? You mean the network itself, or is it what is it traced to? By receptor densities, what do you mean?
So on the left-hand side, this is what we get from our machine learning model, right? And the right-hand side, this is some kind of, again, what we can call as average data. So there is a database where someone, let's say, collected all the neurotransmitter data that is available publicly. And there is a toolbox actually, again, created from our institute called USpace. And in that, let's say, in this, we have about 10, 20, but now it has expanded to about 30 different receptors and neurotransmitters. So what you have is a 3D volume. And for each point in this 3D volume, you have the density of this receptor or transmitter as collected by the PET imaging, right? And what we do in this case is we are doing spatial correlation, because for each point or some kind of location in the space, we have the nodal importance scores from the machine learning model. And here we have the densities of the neurotransmitter system. So we take all these values and simply correlate them with each other, right? Okay. So let's move on to the machine learning part. Let's see. So what we were interested in is confounding effects. So I will try to explain very quickly what we mean by that. So these are some kind of signals that we don't want the model to learn. And an example is, let's say, older people are more likely to be diagnosed with Parkinson's, right? So we know that as a fact. So if you have a database of people at different ages, we want the model to learn Parkinson's and not the aging signal. We don't want the model to say old equal to Parkinson. And many models will try to do that because it's a relatively easy signal to learn, right? So aging signal is much easier to learn than the Parkinson signal. So we want to get rid of the aging signal and want the model to focus on Parkinson's signal. So in that case, Parkinson's status is the target that we want to predict, and age is the confound. And the problem, how to deal with this is, again, there are many ways. One kind of very commonly used way is called as the feature-wise confound removal. So what is done is simply you take the confounds and you regress them on each feature one by one. So you build a linear model. Okay. All right. Sorry about that. I hope it continues. So I just minimize this. Okay. You can still see it? Yes. Good. So you simply build a linear model, and these are the 3 steps that you can see there, right? So this is kind of R notation. If you're used to this, you build a model to predict the one feature, a given feature, using the confounds that you have, and then you take the residual of that model as the confound removed signal. So that is XCR, so confound removed. And again, there are some kind of nuances that one needs to take care of when building a machine learning pipeline, let's say to avoid data leakage when you do cross-validation and so on. And we show actually in one of the papers that actually this is very important. And of course, there are other ways of doing this, but this is one of the dominant ways of removing confounds. So we wanted to focus on this particular case. Other possibilities, for example, you stratify. If there is, let's say there is a binary case, or you kind of try to match the distributions in some way. So here is an example how confound removal works, right? Again, on the x-axis, you have the actual hand grip strength. So it's simply kind of a measurement device where you press and it tells you what's the kilogram hand grip strength that you have. And the y-axis is the predicted values. The features that we used is, again, some kind of similar features that I talked about before of the brain structure. So this voxel-based morphometry, gray matter volume kind of features, right? And you can, if you look at the plot, its correlation is 0.6. This is quite high. Like it's a very relatively high number compared to anything else that we have seen so far, right? At least in this study. Age is relatively easy, but for cognition and so on, right? We would think that this is a very nice signal. But what happens if we separate the males and females, right? So if you build separate models for males and females, they don't work as well. So in this case, gender is a relatively strong confounds. So the predictions are simply driven by sex of the subject in this case. So here that is a confound. So now the interesting thing is the accuracy decreases after confound removal. And that basically says the signal, the features were confounded, right? So relatively straightforward, no problem. Now let me show you another case, right? In this case, again, I will try to explain very quickly. What you see is the blue is the confound removed features here. And the orange is the original features. So confound removed features show higher accuracy. So this is Pearson R value on the y-axis. So they show higher accuracy than the original features. So before, what we saw is the accuracy goes down. It's a simple, easy interpretation. But the accuracy goes up. Why did the accuracy go up? Is it the right reasons for the accuracy to go up, or is the wrong reasons for the accuracy to go up? So again, it's the machine learning people are typically pessimistic. We see lower accuracy, we don't complain. We see higher accuracy, we need to know why there is a higher accuracy, right? Okay, good. So we see higher accuracy, it's like, okay, where is the signal coming from? Why did the accuracy increase? Okay, now what we did, so here on the left-hand side, what you see is the XG boost. It's one of the kind of very commonly used popular algorithms, which is based on decision trees. And it's a nonlinear model. So it can model nonlinear relationship between the features and the targets. We repeated exactly the same experiment or the same data with a ridge regression, which is a linear model. And we don't see this trend. We see the blue bar is much lower than the orange bar. So what is happening here? Where is the signal coming from? So that's where we wanted to look into detail. So I will try to give you one more visual interpretation of this phenomenon, right? So here in this example, again, I will not go exactly into details of what white matter disconnectivity is, but this is a 3D kind of projection of a high dimensional data using UMAP. So it's kind of one of the nonlinear dimensionality reduction techniques. If you have heard of this, it's very nice, I think. And the confound we were interested in age. So how old that person is. Again, this is MRI. All of this is coming from MRI data set. And if you just plot this visually, we could not see a very strong signal, right? Then we removed age from exactly the same signal, did UMAP, look at 3D. Now you see a very clear signal. Younger go to the top right, and bottom left is the older, right? We see relatively strong signal of age after confound removal. So it's actually doing the opposite of what it is supposed to do, right? So that's the problematic part. So it's introducing information it's supposed to remove, right? And here is, again, the same plot. In this case, what we did, we permuted the features, right? So there should be no relationship between the features and the target and the confound and the features because they're all permuted now. We see the same signal. So this procedure is introducing the signal even in completely random data, right? And that is very problematic. And I will show you one more case of this. So in this case, we have a very nice clinical colleague who is interested in diagnosing ADHD. That's what he does day in, day out. That's his job. And there is unfortunately no kind of objective way to do this. So everyone wants an objective way to diagnose ADHD. So our idea was we can collect voice features, right? So people are speaking some pre-designed sentences. We collect the voice features. We do standard processing of the voice messages, do some kind of spectral analysis, use them as features, and predict ADHD yes or no, right? Relatively straightforward. There are tons and tons of papers doing that in the literature. So we said, okay, let's do this, right? So why not? And in that case, the problematic part is depression is a very strong confound of ADHD. So it's a comorbidity. So if people have ADHD, there is a strong likelihood that they also have depression. So we want the model to learn ADHD, but not depression. Again, the similar problem, right? So, but there are other possible confounds as well, like education level, age, and so on, right? So what we tried, what you see on the right-hand side is on the x-axis, you see gender, education, age, BDI, and all of them together. So these are the confounds. So we use them as a confounds to do the feature-wise confound removal. And what you see, or you will see soon with the star, is the highest accuracy of them all. So if you remove BDI, after confound removal, so that's the red. That's the red bar there. We will get the AUC of 0.9. It's super high. This can be, if this is true, this will be a clinical diagnostic tool, right? Everyone wants that. But again, we ask the same question. We see a very high accuracy. Is it for the right reasons? And then we again did all the analysis as I explained before. And indeed, we see that no. Unfortunately, that's not the case. It's a little bit tricky to explain the plot right now. But here again, we shuffle the features, right? So for simplicity, all we did is shuffle the features and redid the machine learning analysis. Again, you see the red bars, they're quite high. There should be no signal in the features. They are completely permitted of their original version, yes, but they're permitted features. We still seek a very high accuracy. So the model is learning something, but it's learning it for wrong reasons. And we call this phenomenon confound leakage. Because in the process of confound removal, we end up introducing information that we should have removed. And I hope with all these three examples, well, two examples that I showed you, and the third example is under construction, I hope that I managed to convince you that this can happen and probably has happened in many machine learning pipelines. What we see is nonlinear models, let's say random forests, or something that's based on a decision tree, or some kind of neural network-based models. They are much more susceptible to this phenomenon than linear models. And it kind of makes sense because they are very good in picking up the signal. Even very tiny bit change in the distribution, these models will learn it. And because the confound has that information it's introducing, even a slight shift will be detected by these models. And if you want details, we have the paper on archive, which is currently under revision as well. So we try to give at least some explanation. Of course, there could be other possible explanations. We cannot explain it all, but at least we try to explain some of it. And we think it's because of the skewed distributions, which are very difficult to get rid of in almost any application. I'm coming towards the last few slides that I have for today. So this is the confound leakage is just one concern, which is much more pertaining to the machine learning part of this analysis. But there are many other problems and that come up again and again. And I've been fortunate enough to participate in many of the studies that kind of are going in this direction. The first one is the low prediction accuracy. It's a big, big problem, right? And we want to know how brain creates behavior, but it's very difficult to get there. So we wanted to know exactly what the limits are. And that's again where we're trying to go. And this is the first preprint that you see on top right. Again, we show that the low accuracy, but a lot can be explained simply by the demographics. So again, why to do this very expensive MRI scans if just the demographics, which is very easy and cheap to get, they can explain a lot of variance, right? Again, we want to, of course, understand the brain behavior relationship, but where is the signal coming from? And then all the confounding and the leakage problems come in that case. Right. The second is a lot of these behavioral measurements are not reliable. They're not reliable enough. And what does that mean? If you measure the same person twice, there will be some variance. And sometimes this variance is too much. And of course, we cannot predict it because it's not reliable. So that's another possibility. And we also have this paper under review right now, where we actually try to get into details of how that affects machine learning pipelines if you have unreliable predictors, unreliable targets. In predicting those, what happens exactly then. One of the questions there was before, again, what was the ethnicity, for example? And this is a paper that came out, I think, last year or the year before, the data biases. And we actually show, or, well, I'm part of the author list, so that's why I say we, but they actually show, is if you build behavior prediction models, for white Americans and African Americans, they behave very, very differently. And that's a big problem. And because the sampling issues, because of the availability of the data and so on, we don't even have enough data of the Indian subcontinent MRI data, where we even try to do something like this is currently very, very difficult. So that's something that I would really like to look into the future, right? So is there enough data to collect and to see if we can create good processing pipelines and then prediction pipelines to use this type of data? And the last one, but again, not least, is replicability and analysis freedom. So here, again, we were fortunate enough to participate in a study. It's like there was a single neuroimaging data set that was analyzed by many different teams. And our group also participated in that. And they show that the variability is just too much. And again, this is because of the design choices, of the analysis freedom. Probably people have feedback loops internally when they do the analysis and so on. So the replicability just drops because of these problems. So all of this needs to be taken into account to move forward to actually make use of not only MRI data, but I'm pretty sure this type of problems appear everywhere in science. Right? But again, kind of a little bit positive note, right, towards the end. So again, there are technical implementation challenges, for example, pre-processing of very large amounts of data. So for that, we use something called as the data lab. So which is a very nice, again, tool created by our colleagues, which has very nice data provenance tracking and so on. Also avoiding data leakage, it sounds reasonably straightforward, but implementing the software that does this correctly is a big challenge. So we have created a software library, again, following the name of our research center. We have started many things with JU. It's a ULearn library, which makes many things easier for you. So if you're interested in doing things like what I just showed, have a look at this. Again, feature extraction, right? So all this kind of VBM features, the connectomes and so on. So we have also kind of tried to standardize this into another library. And these are some kind of details. The nice thing is both the libraries are modular and extendable. So we provide some kind of things that are widely used in the literature. But if you want something very specific, you can easily write your own transformer. So this is kind of building, at least ULearn is building on top of scikit-learn. So if you have a scikit-learn compatible transformer, you can more or less plug and play. And with a few lines of code, you can do nested cross-validation and kind of get the correct output and inspect the models and so on. So it provides many kind of additional functionality that you can easily get without coding too much. And similar things are also for the feature extraction pipeline. We are trying to standardize them, but also keep it flexible enough so everyone can kind of extract what they like. And of course, this type of work is not possible without help of many, many people, many good people. So you see a few faces from the group that we have, the applied machine learning group in Juelich. Of course, many, many collaborators that have contributed to this work. I have been lucky enough to work with this kind of very diverse team. And of course, without funding, nothing happens, right? So we were lucky enough to also get a reasonable amount of funding to keep things going. So thanks to them, and also thank you to you for paying attention and all the nice questions, and I look forward to more questions. Okay.
Okay, Kaustubh, excellent. Thank you so much for taking us through the many areas which are of course interlinked, but also posing quite a few outstanding problems. I hope some of the younger audience are able to kind of lap onto those. Okay, fine. Are there any questions from audience here? Here.
So how do you get access to clinical data? I mean, are there hospitals which are involved in your collaboration?
Yes. So there are two ways. So there are some clinical data which is accessible upon registration. For example, the Alzheimer's data that I showed, it comes from the ADNI database, which is again one of these very long-running collaboration efforts, mostly in the US. And you need to go to their website, register, and give a relatively short research proposal, and they evaluate it. And then they provide you access that you can just download and use that data set. So that's one way. And there are many other data sets like that that are accessible, in some way openly accessible upon registration. And we also have collaborators. Of course, in the University Hospital in Düsseldorf, we work very closely with them, but that region is quite dense in a way. So we have access to data from many university hospitals like Aachen, Cologne, Bonn, and even beyond that, even collaborators in the US, we have quite much data from them.
Kaustubh, this is a non-technical question by the time somebody asks. So I understand that you are in collaboration with the IIT Bombay group, and you are part of setting up a center for these studies. So how do you think, for example, of course, we are co-located, there are lots of students from life sciences in this campus, and also quite a few of faculties who work machine learning techniques, not necessarily applied to the topic that you said, but to the other fields. So do you see a kind of synergy or future development that can happen?
Yes. Definitely, yes.
Okay.
So of course, machine learning it's a toolbox, right? So, and we want many machine learning methods that work reasonably well on small amount of data. So that's kind of one of the biggest challenge, right? So if you have tons and tons of data, it's relatively easy to build models if there is a signal, right? But with the small amounts of data, it's very difficult. So of course, this is one of the points where I think we can do a lot, and there is a lot to be done still. So this is one focus point, but of course there is also ideas, right? So not only in the machine learning direction, but exactly how to approach the problems and how to solve them, and the domain-specific problems. So there is definitely a lot of kind of cross-linking that can happen. And additionally, I think we are always looking for good people, right? So I can also say that very openly. So if anyone is interested, please contact me directly, so I can show my email address again if you like.
Sorry, I see Sushil wanted to ask you a question.
I have a question, maybe not directly related to your research, just generally speaking. If we have a person who we think is beyond his age in terms of thinking, who would probably lie at the age of 20, his brain is, let's say, 25, 26. Okay, if it is that way, is it known as the person grows old, if he still remains above the median crowd, or is there any information on that?
That's a very interesting question. At least I have not come across any studies like that. So I know a couple of studies that actually show that in some cases, the brain looks younger than it should. And this correlates reasonably well with certain behavioral/lifestyle choices. So there are a couple of studies that go in that direction, but I'm not aware of studies that actually focus on what you mentioned in terms of, let's say, the trajectory, longitudinal trajectory. So I'm not aware of studies like that.
Thank you.
I
I can ask you.
up to you. I mean, I'm happy to.
Basically, what are the predictor biomarkers that one would expect? Because then this would, from an industry point of view, becomes very translatable to application in healthcare.
Mhm.
So what are the predictive biomarker sort of regions, like prefrontal cortex versus amygdala versus
Yeah.
different regions that you think would be the hub for doing like directed small work directed, but which will result into faster diagnosis from that point of view?
Yes, very good question. In some cases, we know that, right? Or at least we have some reasonable idea. Let's say you have, for dementia, Alzheimer's direction dementia overall is hippocampal volume, right? So that's reasonably known. So if the hippocampal volume goes down, that we know that there is going to be some kind of signal in that. So and that is being used to quite some degree,
Yeah.
as far as I know. But for many other issues, so Parkinson, there is some kind of idea, but not as well known yet. And for psychiatric disorders, it's very, very difficult.
Yeah.
So there are, again, some kind of indications, let's say fear centers or some kind of basal ganglia system. So it's known that they might be involved, but the biomarker of that level, which can be actually translated, is so far, at least, I'm not aware of.
Yeah.
Right.
Also, because a lot of these regions are subcortical, and
Yes.
the data that we get from fMRI subcortical would be very, so an
Yes.
implementation of better machine learning techniques to, remove the noise from that data would be very cool.
Definitely, yes. But if you're interested, we can talk in a lot more detail afterwards.
Yeah, that is, I think, maybe a question for our tea. You can continue discussing. So once again, I think it's a great pleasure once again thanking today's speaker for really bringing up such a nice topic and also elaborating us to our level.
Thank you for having
I
me.
wanted to kind of tell you that the forum in which you are speaking today has completed 40 years of its existence. So we have kind of made a memento for you, and it's great pleasure
That's
giving
nice.
one to you on this occasion.
Thank you.
Thank you.
Looks
Thank you
very
so
nice.
much. Thanks. Yeah.
Mm,
I'd love
yes.
to give you. Okay, so that brings to the end of this lecture. So please join for a cup of tea in the West Canteen.